Hello and welcome to our video lecture series on Chapter 7. This is Part 2. Part 1 was lectured on in person, so we are going to start with Section 7.5 on metabolism without oxygen. And our learning objectives here are to discuss the fundamental difference between anaerobic cellular respiration and fermentation, and describe the type of fermentation that readily occurs in animal cells and the conditions that initiate that fermentation. So we'll start off with anaerobic cellular respiration. Anytime we see this word anaerobic, it means without oxygen. And so we're going to see a lot of organisms within the domains of bacteria and archaea that are going to utilize anaerobic respiration. An example would be the group of methanogens, which are archaeans that produce carbon dioxide to methane to oxidize NADH back to NAD. And they're going to be found within soil and in cow and sheep digestive tracts. Another group is the sulfate-reducing bacteria. They reduce sulfate to hydrogen sulfide to regenerate NAD from NADH. And you could see in this image over here, this green area within these coastal waters is showing that there was an eruption of hydrogen sulfide due to this type of bacteria producing it. And they release it as a gas as they decompose algae in the water. So let's talk about lactic acid fermentation. Fermentation is a method that is used by animals and certain bacteria like the ones we find within our yogurts. And we see this process within our red blood cells since they lack mitochondria, and within our skeletal muscle when the oxygen is low. So perhaps we have been working at really high intensity or for long durations of time where the oxygen is not sufficient. So at a point, lactic acid is going to accumulate within our muscle and starts to get removed and placed into our bloodstream. That point in time is called our lactate threshold. And if our lactic acid loses the hydrogen ion, lactate then is going to be brought over to our liver for metabolism, where it can be reconverted to pyruvic acid. And you could see this process taking place over here. Of course, glycolysis is going on here, giving us our NADH and our pyruvate, but we are going to convert that pyruvate utilizing an enzyme known as lactate dehydrogenase over to lactate. Now this usually is a reversible reaction, but it's prevented from going into reverse due to acidic conditions. And it was once thought that lactic acid would accumulate within the muscle and cause muscle stiffness, fatigue, and soreness. Although recent research has found that that is not true, and really the stiffness, fatigue, and soreness is due to a inflammatory process within the muscle rather than the lactic acid itself. All right, let's talk about alcohol fermentation. This process is going to produce ethanol, and it really comes down to how yeast is going to eat a form of sugar, which is our starting point from which beer is made, wine, to sake, to cider, and even the hard liquors like tequila, but they are going to also go through another process called distillation. So we won't go into all of that, but you may be wondering, what really is yeast? It's a tiny single-celled organism, and it is going to work to convert sugar into alcohol, and it can create carbon dioxide and heat through this process. And so when we are doing something like trying to make wine and fermenting grapes to about 60 degrees to 85 degrees Fahrenheit, from that heat that we're adding in, the yeast will start to eat the sugar until it's all gone, and that'll give us an alcohol percentage of about 16%. Anything above that is going to kill the yeast and stop it from converting more sugars. So let's take a look at this equation that we have here. The first, and I'm sorry, my mouse is disappearing on me again. Okay, there it is. So the first reaction is going to be when we take pyruvic acid and a hydrogen ion and utilize an enzyme known as pyruvate decarboxylase. And this is a cytoplasmic enzyme with the coenzyme, which we call it TPP, and that really is a thiamine pyrophosphate that is derived from vitamin B1, and we also call it thiamine. And what we get as a result in our product side is going to be carbon dioxide, which is going to be a gas, and then our molecule that we're left with is acetaldehyde, and we get an NADH and a hydrogen ion. So now we're going to utilize this in a second reaction, and this is going to be catalyzed by an enzyme known as alcohol dehydrogenase. And this oxidizes the NADH into NAD and will reduce our acetaldehyde into ethanol. Now we do have some other processes of fermentation that take place, and this is typically within bacteria. We have a classification of facultative anaerobic bacteria that are able to switch between aerobic respiration and fermentation. Essentially, it really depends on if that oxygen is present. It will utilize aerobic respiration, but if not, switches to fermentation. But then other bacteria are known as obligate anaerobic bacteria, and that obligate tells us that it has to be anaerobic. So in these cases, oxygen is like poison to this type of bacteria. So they live and grow in the absence of oxygen, and a genera of bacteria that do this is Clostridia. And gas is released after fermentation reactions, and it really can help us in the lab identify different types of bacteria based on what gas is released. And lastly, we can just mention that the organisms use fermentation to ensure that they have enough NAD for that sixth step in glycolysis in order to make ATP from glucose.